Good morning. Today, inshallah, in this session, we're going to cover some points that we haven't covered properly or thoroughly in slides 22 to 26. And we are going to start by reviewing enzymes and enzyme properties. You know that mechanical digestion should be followed by chemical digestion. Chemical digestion is the breakdown of complex, insoluble, large molecules into small, simple, soluble molecules, because only these small, simple molecules can reach the blood. Why do we want the molecules to reach the blood? We want the molecules to reach the blood in order for them to reach the cells, in order for the cells to be able to break down those very simple molecules or relatively simple molecules relative to the original complex molecules, break down and extract the energy in the bonds by means of a process of cellular respiration. So chemical digestion is essential for arrival of food to the blood. And arrival of food to the blood is essential for arrival of food to the cells. And arrival of food to the cells is important to carry out cellular respiration and release all the energy, or a great part of the energy stored in the bonds of the food molecules, for making use of this energy for cellular activities. Chemical digestion occurs, as you know, through the process of hydrolysis, which is the breakdown by the addition of water, and it requires very specific molecules that are called enzyme molecules. All this we have covered in the classroom in the previous class. And now we are going to focus more on the slide of absorption, because we need to review some important concepts like the final products of absorption, where we get those final products of absorption, how the wall of the small intestine and the wall of the blood capillaries are well adapted to carry out its function. So here, this is a diagram that shows you the endothelium, or the endothelial cells that lie the small intestine, that line the small intestine from inside. So these are the endothelial cells. And since they belong to the small intestine, we call them the intestinal endothelial cells. And towards this direction, we have the lumen, or the cavity of the small intestine. So here we have the small intestinal lumen or cavity. And in the small intestinal lumen or cavity, we have multiple types of foodstuff. We have two major groups. We have carbohydrate molecules and protein molecules. These are, as you see, relatively large molecules. Of course, this is not the whole molecule. What's on the diagram is only part of the molecule. And these complex molecules will be converted into their simplest form. The simplest form of carbohydrates are the simplest sugars, or the monosaccharides, for example, glucose. So glucose is a good example and the most abundant example of monosaccharides, and proteins will be digested into their final product, which is the amino acids. So what are the final products of digestion of all foodstuff? The final products of digestion of proteins, after having digested them completely, are the amino acids. And by the way, we call them amino acids and not amino acids. The final products of digestion of carbohydrates are the monosaccharides. And we call them monosaccharides because they are formed of one molecule, one block. There are no repeated blocks inside the molecule. We are going to discuss the names of monosaccharides later on during our detailed study of digestion in the small intestine. And as we said last time, the final product of digestion of the fatty acid, the final product of digestion of fats are fatty acids plus a very important substance that is called glycerol. These final products of digestion of all foodstuff are obtained in the terminal part of the small intestine. As we said before, all final products of digestion, like the amino acids, like fatty acids, like glycerol, like the monosaccharides or the simple sugars, are obtained in the last part of the small intestine. So where do we get all these final products of digestion? We get them in the ileum, which is the terminal part of the small intestine. And now we're going to see how the endothelium, or the cells lining the cavity of the small intestine, are well adapted to carry out their function. So if you look at this border, we call it a brush border. It looks like a brush. Why do we call it a brush border? Because as you see, there are tiny projections that arise from this border. And by the way, it is the border that lies towards the lumen of the small intestine and towards the site where the final products of digestion are present. And that makes sense because if you need to absorb those nutrients, you have to provide a large surface area for digestion. These projections, these tiny finger-like projections, are called villi. The singular is villus and the plural is villi. So what are villi? How do we describe villi? We describe them as tiny finger-like projections. Tiny because they are very small, finger-like because they look like fingers, and projections because they protrude, towards the lumen of the small intestine. What's the importance of those villi? The importance of those villi is to increase the surface area that is allowed for absorption. Increase the surface area. So the more villi you have, the more the rate of absorption. And on the other hand, if you have an autoimmune disease or a medication that destroys those villi, then you are going to have difficulty in absorption or bad absorption, which we call malabsorption. Because you know that mal in French, or mauvais, means bad. So malabsorption is bad absorption because of absence of adequate amounts of microvilli. And now we come, the last point on that slide is the definition of absorption. So we are going to move on to the definition of absorption. Absorption is nothing but the transfer of those final products of digestion from where? From the lumen of the small intestine to the lumen of the blood capillary, because here we have the blood capillary, but we have just shown the cells that line the blood capillary. So here we have endothelial cells that line the blood capillary. So when you transfer the soluble products of digestion from the lumen of the small intestine to the lumen of the blood capillary, then you are carrying out absorption. And during the journey from the lumen of the small intestine to the lumen of the blood capillary here, those simple molecules have to pass through two kinds of endothelia. The first endothelium is the endothelium of the small intestine, and the other one, those small simple cells, are the endothelium of the blood capillary. And this is what's written at the bottom of the slide, where you can see that this passage of food molecules occurs through the endothelium lining the small intestine and then the endothelium lining the blood vessels. So what does this mean? This means that if this is the ileum or the last part of the small intestine, then next to it you have a blood vessel or a blood capillary. So the ileum is richly supplied by blood capillary in order to allow absorption. And here the endothelium consists of tall cells with tiny finger-like projections, whereas here the endothelium consists of flat, small cells like these. They look like this, and they have to be thin in order to facilitate absorption, in order not to increase the barrier that is offered to absorption. We have covered, of course, or we have explained many of the properties of enzymes. We have said that enzyme molecules are protein molecules, and these protein molecules are 3D molecules. Why are they 3D molecules? Because they need to have an active site, and the active site is the place where the specific reactants will bind. So here, for example, if this is substance A, that needs to be broken down into B and C, then A has to fit here. If it does not fit into the active site of the enzyme, then it will not undergo the chemical reaction rapidly. So the active site is the place on the enzyme molecule whose shape is complementary to the reactant and that can fit specifically with the reactant. So why do enzymes have 3D configurations or shapes? In order to allow them to have the active site. And we said that they are biocatalysts. They are catalysts because they speed up the chemical reaction, and they are biocatalysts because they act inside the cell. And we also said that they are used in very small quantities because they are not consumed in the reaction, meaning that if you have an enzyme molecule, so substance A fits with the active site of the enzyme, and then substance A is broken down into, for example, the two products, which are B and C. And then after this has been done at a very fast rate because of the presence of the enzyme, then the enzyme will unload itself. It will unload itself from B and C. B and C will leave the active site, and the active site of the enzyme is now free. This active site, this unloaded active site, is free again to receive a new molecule of the reactant A in order to start or speed up a new chemical reaction. Okay, so enzymes are not consumed in the reaction. That's why you can use them over and over again, because since it's not consumable, then you can reuse it again. And since it's not consumable, you can just use a very small amount of the enzyme because it will not disappear from the medium of the reaction. We also said that enzymes are specific. Why are enzymes specific? Because every enzyme has an active site that has a specific shape, and the specific shape can fit with a specific reactant. So their shapes are complementary to each other. So the enzyme and the substrate are complementary in shape to each other, and that's why they are capable of fitting with each other. And what is the substrate? The substrate is the reactant that can fit with the enzyme, or the substrate whose shape is complementary to the shape of the enzyme. Since only one substrate or very few substrates can fit with one enzyme, then the enzyme is specific, then every enzyme can catalyze a specific or particular chemical reaction. Most enzymes catalyze only one chemical reaction. We have covered all this, and this was a necessary introduction to focus now on two new concepts, which are how enzymes speed up chemical reaction. Enzymes speed up chemical reaction by lowering the activation energy, which is the minimum energy needed to start a chemical reaction. And the second concept on that slide that we need to elaborate more on is the old and recent theory. Both theories try to explain enzyme action, but one of them is now rejected and the other proved to be correct. We will start by the theories, and there are two theories to explain enzyme action. The first theory is the lock and key theory. The first theory is the lock and key theory, and the second theory is the induced fit theory. Induced fit theory. So the first theory is the lock and key theory, and the second theory is the induced fit theory. In the lock and key theory, we assume that the substrate, which is the reactant, the substrate, which is the specific reactant. So here we have the substrate. You can just use a very simple synonym for a substrate, or a very simple meaning definition for the substrate, which is the specific reactant. And specific reactant is the reactant that can fit with the active site. The specific reactant has a shape that is complementary to the active site. That's why it is the only one that can fit with the active site. The lock and key theory exaggerates that fitness and that complementarity and assumes that this molecule, which is, for example, molecule A, fits perfectly. So this theory assumes that there is perfect fitting between substance A, which is the substrate, and the enzyme molecule. Just like a lock and a key. For every lock, you have only one key. Why? Because every key should fit perfectly with the opening in the lock. Otherwise, you can open, for example, your friend's lockers or your classmate lockers with your key. And this destroys the function of a key, by the way. But the induced fit theory proved to be more reasonable, because the induced fit theory assumes that there is imperfect fitting between the enzyme and the substrate, but it is an almost perfect fitting, meaning that, for example, if this is the enzyme molecule and that is the substrate molecule, as you can see, they are not exactly fitting. But this molecule is the most perfectly one, or is the most closely fitting with the enzyme. So what happens when the substrate A tries to fit with the active site of the enzyme is that this fitting, which is not 100% perfect, will induce, meaning that it will trigger or stimulate changes in the shape of the enzyme. So it will trigger shape changes. Where? In the substrate? No, in the enzyme molecule, so that the enzyme molecule becomes more fitting, so that the enzyme and the substrate now fit perfectly with each other, and that's why it is called the induced fit theory. So this is the difference between lock and key theory and induced fit theory. The lock and key theory assumes perfect fitting. The lock and key theory assumes perfect fitting. This is the first difference, but the induced fit theory assumes an almost perfect fitting. So it's only a word that makes the difference, an almost perfect fitting. In the induced fit theory, the shape of the enzyme or the enzyme shape does not change. The enzyme shape does not change. But in the induced fit theory, the enzyme shape changes a little bit. So these are the major differences between the nowadays rejected lock and key theory and the induced fit theory. And now we're going to talk about the second point, which is how enzymes speed up chemical reactions. How enzymes speed up chemical reactions. How enzymes speed up chemical reactions. In order for every chemical reaction to start, whether it is an exothermic reaction that releases energy or an endothermic reaction that absorbs energy, you need to give a certain amount of energy. You need to provide the reaction. For example, if you have a test tube, you can just heat the test tube. So heat is one type of energy that you can provide to the reactant A to be broken down into B and C. So you need to provide energy, whether this energy is heat energy or electric energy or like an electric spark, for example, or whatever kind of energy. The minimum amount, the smallest amount of energy that you provide to help the reaction start is called the activation energy. So what is the activation energy? The activation energy is the minimum amount. Why do I have to say minimum amount? Because if 10 joules of energy are enough to start the chemical reaction, then definitely 15 would be enough. But maybe if you increase more, the molecules would be damaged by the heat energy. So activation energy is the minimum amount of energy that is required to start the chemical reaction. Say, for example, that providing 10 joules of energy takes 10 seconds. Then maybe providing 100 joules of energy would need 100 seconds, and therefore it would need a longer period of time, and this would slow down the reaction. So it makes sense that enzymes that are by definition molecules that speed up chemical reaction would definitely want to lower the activation energy. For example, if the chemical reaction, as we said before, chemical reactions can occur on their own, can proceed on their own, but very slowly. So if the chemical reaction, when proceeding on its own, requires, for example, 100 joules to proceed and to start, then if you use an enzyme, it would not need 100 joules, but it would just need 10 joules of energy. So what the enzyme did is lowering the activation energy, and by lowering the activation energy, then it speeds up the chemical reaction because it minimizes the time needed to start the chemical reaction. So if I ever ask you about the relationship, between the activation energy and between the speed of chemical reactions, then you would say that as we diminish the value of the activation energy, the speed of the chemical reaction would increase, and therefore the reaction is inversely proportional. The reaction is inversely proportional. Okay? This is one question that you will encounter on your homework assignment sheet. And now we are going to ask another important question, which is how do enzymes lower or diminish activation energy to speed up chemical reaction? How do enzymes lower activation energy to speed up chemical reactions? As you know, that molecules that react with each other, say for example, A and B react together to form molecule C. So in order for A and B to react together, they have to collide, they have to collide with each other. They have to collide with each other. And in order for them to collide with each other, they have to have kinetic energy. So molecule A should have kinetic energy A, molecule B should have kinetic energy. And as you know, kinetic energy is the energy that accompanies movement. So if molecule A has good or enough kinetic energy and molecule B has enough kinetic energy, then they might collide with each other. Collision is very important for two molecules A and B to collide with each other. And not only to collide with each other, but to collide with each other in the proper direction, meaning that there is a certain direction in which A and B have to collide with each other, direction, in order to react with each other if they are capable of reacting with each other, if their electrons and their valence electrons allows them to collide with each other. So how does this relate to energy? The more you give of kinetic energy, the more the possibility of molecules colliding with each other, just like when you have students in the playground, if they're just walking or standing up quietly, they wouldn't collide with each other. But if they are running fast, then there is a higher possibility for them to collide with each other. So the more you give of kinetic energy, the more you have collision between the molecules and the more the possibility for them to react. What's the role of the enzyme in helping collision of the molecules in the proper direction? Meaning that if the necessary condition for molecules to react with each other is them colliding with each other, or is them colliding with each other and in the proper direction. So if you can achieve this purpose of colliding with each other without increasing energy, then you did a great job, because you would help reaction of the molecules without the necessity of increasing energy. And this is exactly what the enzyme does. This is exactly what the enzyme does. If you look here, this is an enzyme molecule. This is the active site for molecule A, and that is the active site for molecule B. What the enzyme does is that through having those active sites, it helps lodging of the molecules in their proper places. And not only does the enzyme help them come closer to each other, but closer to each other in the proper direction. So that's why the enzyme saves energy. That's why the enzyme diminishes the activation energy, because it allowed collision of the molecules in the proper direction without being forced to give them energy, just by doing what? What does the enzyme do to achieve this? Just by approximating them together, just by getting them in close proximity to each other, closer to each other in the proper direction, so that they can collide. Once they collide, if they are liable to react with each other, then they would give the reactant. And after having given the after having given the reactant is unloaded, or the enzyme unloads itself, and the enzyme returns untouched. The enzyme returns, and by untouched here, I mean unaffected, I mean unbroken, and that's why it can be reused again and again. And this is the relation between enzymes and activation energy. Now, the last thing to say here on slide 25, I think, is that every enzyme has an optimum temperature at which it works the best. Why? How does enzyme temperature affect enzyme activity? We know that the enzyme has to have a specific shape to accommodate the substance, the two reactants that we have just seen on the previous slide. If you have drastic changes in temperature, like when you are feverish, for example, then the enzyme molecule will change its shape, and it would be no more capable of fitting with the or accommodating the substrate. For example, if the temperature increases too much, then the shape of the active sites would be like this. The enzyme molecule would have changed its shape, and now molecules A and B would no more be able to fit with the enzyme action. So every enzyme has an optimum temperature at which its activity is the best, is optimum. Any shift above or below that optimum temperature will result in a diminution in the shape, in the activity of the enzyme. For example, here, this slide shows optimum temperature, and it is the temperature at which the activity of the enzyme is maximum. For our human body, because our human body has a more or less stable temperature at 37, then the best temperature for the enzyme, for all enzymes in the human body, is 37. Here, this curve shows you the relationship between the temperature, which is the independent variable, and enzyme activity. We always put the independent variable on the x-axis, as you know, from your study in the middle school, and we put the dependent variable on the y-axis. So whenever you change the independent variable, which is here the temperature, and you have to put the unit on the graph, the dependent variable will change, and that's why we call it dependent, because it depends on the changes in the independent variable. So in this graph, we can see the relationship between the temperature in degrees Celsius on the x-axis and the enzyme activity on the y-axis. As you can see, there is a peak for the curve, and if you drop a perpendicular, you will see that it will meet the x-axis at 40 degrees Celsius. Of course, our optimum temperature is not 40. Our optimum temperature is 37. So this might be a graph of another living organism whose optimum temperature is stabilized at 40. If this were a graph that represents enzyme activity, the relation between temperature and enzyme activity in a human being, then when we drop a perpendicular from that maximum activity of the enzyme to the bottom, it should reach 37 degrees Celsius. Okay, so this point at the peak of the curve represents what? It represents the optimum temperature of that particular enzyme in that particular living organism. How can we define the optimum temperature? It is the temperature on the x-axis that corresponds to the maximum Y value, or that what is the Y value here? It is the rate of the chemical reaction. But what happens when we are around the optimum temperature? What happens when we go farther from the optimum temperature? If this is the optimum temperature for that particular organism, then when you raise the temperature, what happened to the enzyme activity? The enzyme activity dropped. And what happened on the other side if you lowered the temperature? The enzyme activity also dropped. So the optimum temperature is the temperature at which you have maximum activity. If you go more than the optimum temperature, enzyme activity will diminish. If you go lower than the optimum temperature, enzyme activity will also drop. So you have the best or the optimum enzyme activity when you are at 40 here, which is the optimum temperature. Why does this occur? If this is the optimum temperature, then above the optimum temperature, if you reach a very high temperature, like 60, for example, you can see that at 60, there is zero enzyme activity. Why? Because the molecules of the enzyme are deformed. The molecules of the enzyme are deformed, and therefore they can no more fit with the substrate. The substrate can no more fit with the enzyme because of the change in the active site, meaning that if the active site was shaped like this, for example, if the active site was shaped like this, and substance A was capable of fitting with the enzyme active site, if you heat at 60 degrees Celsius, which is the highest temperature here on the x-axis, then the shape of the active site and the shape of the enzyme molecule would change, and A is no more capable of fitting with this deformed active site. In such a case, we say that the enzyme has been deformed or the enzyme has been denatured. So denaturation occurs when there is excessive heat. And when there is excessive heat, the molecule of the enzyme has some chemical bonds that are broken inside it, and therefore it changes its shape, because the original genuine 3D shape of the enzyme molecule is stabilized by chemical bonds. If you break down those chemical bonds, you make the molecule messy, and the molecule changes its shape, and it is no more capable of having the good active site, the perfect active site it had before, and the reactant will not be, or the substrate will not be able to fit with it. This is what happens when you increase the temperature. So when you increase the temperature at the maximum, like 60 here, there is denaturation that occurs. There is a deformation or deformity that occurs in the enzyme molecule. You have now a deformed enzyme molecule that is no more capable of doing its function or carrying out its function. On the other side, why when we lower the temperature, enzyme activity diminishes? It diminishes because of a decrease in kinetic energy of the molecules, and therefore a decrease in collisions, and therefore a decrease in the chemical reaction. So any shift away from the optimum temperature will diminish enzyme activity. Above the optimum temperature, when you reach very high temperatures above the optimum temperature, you break down bonds through this very high temperature. You break down bonds in the enzyme. You make the enzyme molecule deformed, and it is no more capable of carrying out its function. If you diminish the temperature, the kinetic energy of the reactants diminishes, collisions diminish, and therefore the rate of the chemical reaction will diminish. Every enzyme has one optimum temperature that is its own optimum temperature depending on the medium where it acts. Human enzymes, all human enzymes have 37 degrees Celsius as the optimum temperature because this is the temperature of our human body. Not only the temperature affects the action of the enzyme, but also the pH affects the enzyme action. I don't know whether you studied pH in the middle school or not, but the pH is, we can just simplify the definition of pH for the time being and say that pH is an indication of how acidic the solution is or how alkaline the solution is. So the more you have of hydrogen ions, the more the solution is acidic. Like when you have a lot of HCl, hydrochloric acid, in a test tube, for example, this is an acidic solution. But the more you have of OH, which is the hydroxyl group, the more alkaline the medium is. Like, for example, sodium hydroxide solution, it's like a soap solution, for example. Soap solutions are alkaline. We will be discussing this more and more in the classroom, but this is just an introduction to the concept of pH, meaning that how acidic or alkaline a solution is. For example, saliva is slightly alkaline because it contains molecules that can give rise to sodium hydroxide. The stomach is slightly acidic, not slightly, the stomach is very acidic because it contains HCl. The small intestine is very alkaline. It is one of the most alkaline media in the human body. Therefore, every enzyme put in a certain cavity should have an optimum pH. For example, since saliva is slightly alkaline, then enzymes that work in the saliva have their optimum pH slightly alkaline. Since the stomach is strongly acidic, then the enzyme that acts in the stomach, which is pepsin, should prefer the acidic medium, and its optimum pH should be acidic. In the small intestine, all the enzymes that act in the small intestine love the alkaline pH. Their optimum pH is alkaline. That's why if in temperature we had one optimum temperature for every enzyme, for example, all human enzymes have 37, so we have one graph, but here we have several graphs. For example, pH 2, this is the acidic pH in the stomach. So which enzyme has its peak action at 2? It is pepsin. On the other hand, for example, salivary amylase, which is the enzyme present in the mouth cavity, has an optimum pH of 7.4. So this is a slightly alkaline, and we will know why next time, inshallah. And for example, 2 is strongly acidic, and that's the enzyme that works in the stomach. So a strongly acidic pH is present in the stomach, and pepsin loves this acidic pH. A slightly alkaline pH is present in saliva, and salivary amylase loves this slightly alkaline pH. A strongly alkaline pH around 9, 8.5 to 9 is present in the small intestine, strongly alkaline, and that's why trypsin enzyme that works in the small intestine loves that strongly alkaline pH. The message I wanted to deliver is that unlike temperature, every enzyme has an optimum pH depending on where it is acting, in the stomach, in the small intestine, in saliva, in the mouth cavity, as you can see on the graph. Next time, inshallah, we're going to discuss pH in more detail, and we're going to explain this diagram to the left. Thank you, guys, and we'll be receiving a worksheet that is related to the content covered in that PowerPoint, in that recording, except for pH. The questions on pH well done as a classwork in the classroom on Sunday, inshallah. Thank you and see you on Sunday.